Hi, online students. This is Mrs. Hansen, your instructor. I got this new webcam, so I thought I might use it to give you a little mini lecture to help you understand the function and structure of neurons, which is a major topic in Chapter 2 on the brain and neuroscience. So the structure of each individual neuron is a big portion of the material that you're learning right now. And I thought I could use some props to help you understand what the function of each part of the neuron is responsible for. So I will start by telling you that each individual neuron does not come in physical contact with any other individual neuron. There are billions of neurons in the brain, but they are all separated by kind of ocean water fluid that is filled with sodium. So keep that in mind. Each individual neuron is separated by small, tiny spaces between the next neuron, the next neuron, and the next neuron. And this fluid is filled with sodium, and that's a very important thing to keep in mind. So the neurons communicate with each other by sending chemical messages, kind of like pooping them out into the ocean, and then those molecules are picked up by other nearby neurons. So they're receiving the information, but not directly, it's indirectly, because the chemical messages are sent into the synaptic gap. That's what we call those individual spaces between neurons. And other nearby neurons absorb those neurotransmitter chemicals and process the information. The neurotransmitter chemicals are either excitatory or inhibitory, meaning that the neurotransmitter is going to either stimulate the nearby neuron to have an electrical discharge or an electrical charge called an action potential, or it's going to inhibit that nearby neuron from having an electrical charge. You can think of it as stimulating a neuron towards getting active or inhibiting a neuron from becoming active. So it depresses the neuron's function or it stimulates the neuron's function. So each neuron has a top and a bottom, as I said, and the top of the neuron are branches, kind of like a tree. The branches of the tree are called the dendrites, and the dendrites' job is to absorb some of those neurotransmitter messages that were sent into the synaptic gap by other nearby neurons. So I'm holding up a sponge to use an analogy that each individual dendrite, and there could be millions of dendrites on an individual neuron, each dendrite surface is lined with receptor sites. So look at this sponge. A sponge absorbs fluid. Dendrites absorb neurotransmitter fluid. They are individual chemicals. And think of each hole in the sponge as an individual receptor site. Now, the receptor sites on the dendrites can only accept the neurotransmitter molecules that are the exact molecular shape to fit. So it's like individual parking places. So serotonin, the neurotransmitter serotonin, has to find a serotonin-shaped receptor site to fit in onto this dendrite surface. And the same with acetylcholine. Acetylcholine cannot park in a serotonin receptor site. It has to find its own molecularly shaped receptor site, kind of like parking places. So you are responsible for learning, I think, about 7 of the basic neurotransmitters. There are hundreds of neurotransmitters in the brain. If you end up majoring in psychology, you'll go on and take some neuroscience, neuroscience, and you will be learning about a lot more. But we're starting with 7 basic neurotransmitters. Okay, so when a neurotransmitter is released into a synaptic gap, this nearby neuron's dendrites absorb or accept neurotransmitter molecules into the receptor sites that are individualized on the dendrite surface. So there are billions of receptor sites on these dendrite surfaces. And these dendrites then pass the either stimulatory or, stimulating or inhibitory messages to the cell body. The cell body is called the soma. And I'm going to hold up a calculator to represent the function of the cell body, the soma. The soma keeps track of all the incoming excitatory, at which I would use the plus sign to mean excitatory. The soma says, oh, we're getting 5 excitatory messages. Oh, but we're also getting 10 inhibitory messages. And so if you're getting more inhibitory messages than excitatory, then that neuron is further away from reaching an action potential. Action potential is the name of the electrical charge, the electrical current that runs down the axon when it's activated. So a neuron is sleeping or resting. We call it resting when it's not having that electrical charge occur. So that means the neuron is still inhibited and not stimulated enough to become active and discharging an electrical current we call an action potential. So the soma's job is to calculate and keep track of all the incoming excitatory and inhibitory neurotransmitter messages. If enough excitatory messages occur, oh my goodness, look at that. We're just getting tons of excitatory messages. We're getting so excited. This neuron's getting really, really, really excited. And lo and behold, the threshold for having an action potential has been reached. And so now the soma gives the order for the action potential to begin. I'm going to use, where is it? Where's my flashlight? I'm going to use this flashlight as the prop that means an action potential is occurring. So if I turn this, this flashlight doesn't work. Here's another one. All right, so this flashlight, when it's turned on, that means an action potential is occurring. That means a highly charged current is running starting from the cell body down the length of the axon. And what happens is, remember the sodium that's floating in the fluid around the neuron? That sodium is much more highly charged, positively charged, than the potassium that is in the resting potential state inside the axon. So potassium is in there during resting potential, but then during action potential, there's a switcheroo. There's a kind of do-si-do action where ionic gates in the axon open up, and the sodium ions, and I'll hold up some salt here, the sodium ions pass through the ionic gates into the interior of the axon. The potassium leaves. Because sodium has such a high charge, that means it brings this electrical current in, and it travels down the length of the axon in a domino effect. It starts closest to the soma, then it travels down to the end of the axon. It ends up at the axon terminals. And when the action potential reaches the axon terminals, which are like roots of a tree, they are branches as well, but smaller than the dendrite branches. So remember, there's a top and a bottom, dendrites at the top, axon terminals at the bottom. The axon terminals have these storage containers that keep the supplies of neurotransmitter molecules or chemicals, okay? So I am going to hold up a squirt bottle that has the name of a transmitter on here. This one says, you might not be able to read it, this one says dopamine. Okay, so this is a neurotransmitter, dopamine, which is highly involved in our pleasurable activities. When we do anything that is exciting and pleasurable and we want to do it again, we get a huge dopamine rush, meaning that a lot of neurons in our brain are releasing large amounts of dopamine, and of course other nearby neurons are absorbing that dopamine and processing it and then sending more dopamine around to other neurons. So that's when we experience pleasure. So I hope you have a lot of dopamine activity in your life. There are various ways of getting dopamine rushes, but I highly recommend that you stick to the legal ones and the ones that are not artificial chemicals that you're putting into your body or your brain, because it throws off your brain's natural production of these chemicals. Okay, so let me, these reservoirs of neurotransmitters are actually in little sacs called synaptic vesicles, and those are in the axon terminals. Okay, so when the action potential reaches down the axon, and reaches the axon terminals, it stimulates these synaptic vesicles to release. Here, I'll squirt. I'm not going to squirt you, but I'll squirt. I'll squirt. What am I going to, I don't know, I'm squirting my office. You can't see it, but there's actually fluid coming out here. There we go. So that would be the axon terminals releasing neurotransmitter molecules into the synaptic gap so that other nearby neurons can absorb them. And what part of the neurons that are nearby, what part of those neurons would be absorbing those neurotransmitter molecules? Did you name it correctly? What does this represent? This represents dendrites. Exactly. So other nearby neurons, dendrites, would be with their receptor sites accepting different neurotransmitter molecules, either excitatory or inhibitory. And it just goes on and on and on in a huge bucket brigade, transferring either excitatory or inhibitory neurotransmitter molecules along the line. And this tells our brain what to feel, what to, how to move our body, whether we're feeling pleasure or pain, or whether we can inhibit pain. One of the most interesting neurotransmitters that you probably have a lot of experience with is endorphin. Endorphins, let me see if I have serotonin, norepinephrine, I'm looking at all my bottles here, GABA, I'm pulling out everything but, and oh, here it is. All right, so here's a bottle which I have labeled endorphins. Now, endorphins are your natural opiate painkillers. You have natural pain-killing chemicals in your brain called endorphins. When you are seriously injured, for instance, in an accident, have some of you ever been injured and you didn't realize how badly you were injured until later when the pain really set in? The reason you didn't notice initially that you were hurt so badly is because your brain released a flood of natural opiate painkiller, just like heroin, but your brain makes it. So it's the right chemical in emergency situations for your brain to help you survive and either run or fight, because if you're in too much pain, you can't do either one. In the fight or flight response, you need to be able to run and get away from danger or fight. And so the endorphins kill the pain for you a little bit so that you can do a better job of fighting or running. Fight or flight, if you've heard that before, fight or flight response. So our brain has all these chemicals, and my warning to you is do not take drugs that alter your natural brain chemistry, because it'll really screw it up. If you take heroin, your brain, you know, when people shoot heroin or however they ingest it into their body, it is such a huge amount of almost the identical molecular shaped substance. It's so close to endorphin that heroin and morphine can fit right into the receptor sites for endorphins in your brain. That's why it's so effective. But your brain says, wow, we have way too much endorphins happening in our brain. We don't want that much. It's too much. So guess what? Your brain quits producing natural endorphins, wherever it went. Your brain quits producing the natural endorphins. So what happens when the heroin wears off? You don't even have your normal level of endorphins that makes you comfortable throughout the day, unaware of aches and pains in your joints and so forth. So when the heroin wears off, you are really hurting. You are in a world of hurt. Withdrawal is so severe that very few people can resist, you know, the urge to take more heroin, because that's the only thing that's going to kill the pain. It's going to take your brain a long time to get back to a normal level of natural endorphin production, and that's what recovery is all about. I hope this mini lecture has helped you understand a little bit about the structure and function of neurons. I think the one thing I didn't mention maybe is the myelin sheath that wraps around the axon. That myelin is actually fat, so you need fat in your brain. You need a fat brain. The myelin is to speed up that electrical current called the action potential. That fat speeds up the current as it travels. And if you have slow electrical currents in your axons, then you have a slow brain. And if any of you know anyone who has inhaled substances for a long enough period, like sniffing glue or paint fumes or any of that stuff, they're called huffers. You know, if they have a paper bag full of those fumes, then they can ingest those and kind of get high on that. But it actually melts the myelin around the axons. It's do-it-yourself mental retardation. That's what that is. So you were lucky enough to be born with a normal brain, but no, you had to screw it up and melt the myelin, and now you've got a slower brain. And it doesn't really repair itself very well. So you're not going to regrow the myelin you lost by being a huffer, inhaling those toxic fumes. So again, don't do that stuff. Just don't go there, because it really screws up the amazing ability your brain has to produce the right chemicals in the right amount and to pass them along from neuron to neuron in brain circuits. And your brain produces both chemicals and electricity. Remember, the electrical event happens inside the neuron, called an action potential, and the chemical transmission of messages takes place between neurons. I hope this has been helpful, and email me and let me know if this helped you at all, okay? I just got this webcam, and I hope that I can make some more videos to help you with other topics as we go along. Okay, hope you do well on the exam. I'm happy to meet you. I'm sorry I didn't have this set up for week 1. It would have been nice to have a little welcoming video for you. Let me know if this was helpful, okay? And talk to you later.